안녕하세요. 저는 토론토 대학교 셜리우 교수님 실험실에 있는 박엘리야입니다. 오늘 발표는 영어로 진행하겠습니다. 논문 제목은 Brain Microenvironment Remodeling Nanomedicine Improves Cerebral Glucose Metabolism, Mitochondrial Activity, Synaptic Function in a Mouse Model of Alzheimer's Disease. So today's seminar will start with the background about the project and followed by the method, results, and conclusion with the future direction. So Alzheimer's disease is a chronic and progressive neurodegenerative disorder leading to cognitive dysfunction, causing 60 to 70% of the dementia cases. Current diagnosis is heavily dependent on the clinical phenotype using the neuropsychological tests. With the help of liquid biomarkers and neuroimaging, such as PET-CT MRI with the amyloid or tau substrates. The available treatment in the clinic is mostly acetylcholinesterase inhibitors or NMDA receptor inhibitor, which would increase the neurotransmitter in the brain for symptomatic alleviation. However, is not effective in the moderate cases of Alzheimer's disease and is only affecting the symptomatic relief. Therefore, it does not intervene with the disease modification itself. The reason for difficulty finding any disease-modifying treatment for this disease is mostly because of the complex pathology. In the Alzheimer's disease brain, there's the accumulation of toxic proteins such as pathological amyloids or tau, and brain atrophy or neurotransmitter dysfunction, neuroinflammation, oxidative stress, or vascular dysfunction, etc. So even if you treat the disease targeting one of these aspects, such as anti-inflammatory drug, it's not sufficient to modify the entire disease. That's why it's very hard to treat the disease itself. And one more thing to consider is that the disease is happening inside the brain. Brain is very protective organ. It has the blood-brain barrier located between the brain and the body. Therefore, not any substance can easily enter the brain. Therefore, many researchers have designed the strategies to deliver the drug across the blood-brain barrier because 98% of drugs cannot actually enter the brain, even if it's effective. One of the strategies bypassing blood-brain barriers, such as you inject the drug directly into the brain with using very invasive procedures, or you use the nasal route to deliver the drug to bypass the BBB. Or one can use the cell penetrating strategy, which design the drug so that it can have a specific property to penetrate the cells. However, it's very limited in certain cases. Or you can temporarily disrupt the BBB using the mechanical or chemical forces such as heavy ultrasound. However, as you can imagine, it has a side effect and it's not really a safe strategy, although it can actually deliver the drug into the brain. The other strategy, which is relatively safer and effective, is receptor-mediated transport. So on the blood-brain barrier, it has a receptor so that anything that brain needs can be delivered from the blood into the brain. For example, brain needs the nutrients or any chemical hormones to maintain its function. Therefore, if you make the drug that looks like a nutrients or one of them, then it will recognize as a nutrients and transport across this blood-brain barrier so that you can safely deliver the drug into the brain. So this last strategy is what we have chosen to design the formulation for the drug delivery. So just to easily explain about this strategy, um, our nanoparticle is made of polymer and lipid, and we would load the drug into these nanoparticles. And when it's administered systemically, it would bind to the cholesterol, which you can find in the bloodstreams. And once it binds to the cholesterol, it'll look like a big chunk of cholesterol in the bloodstream. Then the brain would see it as a big chunk of lipid and would like to absorb it and use it as energy source. So it's like a Trojan horse where you locate the drug inside the horse and horse would mimic it with the cholesterol. And once it enters the castle of the brain, it can deliver the drug safely for a prolonged period of time. So our platform polymer lipid hybrid nanoparticle is made of polymer, which consists of the starch backbone with polymethacrylic acid and polysorbate 80. And as you can see on the down here, we have loaded the manganese dioxide, which has the therapeutic as well as diagnostic properties. So with KMNO4 and polyvinyl alcohol, you mix them and add the lipid, and you add the amyloid beta antibody conjugated polymer to it, and using the ultrasonification, you produce the nanoparticles. As you can see in this animal data, once you inject them systematically, in this case, tail vein intravenous administration, you'd find the nanoparticle within 15 minutes in the brain, and it provides the prolonged release over a long period of time. So the problem for our case, going back to Alzheimer's disease, was that there was no effective disease modifying treatment available because of the complex pathology, and it's difficult to deliver the drugs into the brain effectively, even if it's working, because of the blood-brain barrier. So we developed a particle called ABTP-MDNP. AB stands for amyloid beta antibody to target the drug into the disease region where the amyloid beta can be found. And the polymer can permeate the blood-brain barrier using ApoE receptor-mediated transcytosis to safely enter the brain. And the manganese dioxide loaded onto this nanoparticle can target multiple pathology using the antioxidative and anti-inflammatory effects, which would further induce any downstream signalings for more therapeutic outcomes. So using the multi-targeted approach as well as brain drug delivery system, we try to develop the disease-modifying treatment for Alzheimer's disease. I'll just briefly touch upon the therapeutic effects of these nanoparticles. So reactive oxygen species hypoxia are found in the Alzheimer's disease, which our nanoparticle is trying to target. Amyloid beta itself can increase the reactive oxygen species by activating certain enzymes, and amyloid beta, well, once it becomes the plaques, it would accumulate around the vessels, which should narrow down the vessels, leading to low blood supply, which would ultimately lead to low oxygen availability. The low oxygen level would decrease the ATP production, ATP is the basic energy source in the cells, and when metabolizing the glucose, and oxidative stress damages the mitochondria. So by targeting these two, we are hoping to reduce the vascular dysfunction, metabolic dysfunction, as well as synaptic dysfunction. To explain further about number 3 and number 4, we can look at the glucose metabolism. So when there is a glucose in the cells, it can go under glycolysis, which is a fast process, but it produces very low amount of energy source, which is ATP. As you can see, just 2 ATP produced per glucose. However, when the pyruvate enters the mitochondria, it can produce much higher amount of ATP, which is 36. However, it requires the oxygen for it to generate this amount of ATP and healthy mitochondria to actually metabolize the glucose. So if you don't have enough oxygen, or if the mitochondria is not functioning properly, with the same amount of glucose, you would end up having very, very low amount of ATP compared to when things are going well. You would have a higher amount of ATP per the same amount of glucose, which would lower the glucose metabolic efficiency. So this is the brief diagram for our research. The nanoparticle consists of the polymer and lipid, and it has the amyloid antibody to target the disease area upon systematic administration, goes to the brain. And under the Alzheimer's disease, it has a low oxygen available, and there is a high oxidative stress, which would damage the mitochondria. So low amount of oxygen as well as dysfunctional mitochondria would lead to low generation of ATP, which is the energy source. In addition, the amount of glucose available is very limited in the Alzheimer's disease because of the vascular dysfunction and all the other pathological consequences. Neurons are particularly highly metabolic cells. Therefore, it needs a lot of energy to perform its function. However, if you don't have enough energy available from all these consequences, it would not have enough function. Therefore, it cannot communicate with each other, which is shown as synaptic dysfunction. But if you treat this Alzheimer's disease with nanoparticle, you would increase the oxygen supply and reduce the oxidative stress. Therefore, the mitochondria would remain relatively healthy. Therefore, it can generate the more higher amount of ATP through aerobic glucose metabolism. And also it can increase the glucose availability in the brain. With all that ATP and energy source, neuron can have a better synaptic function that will lead to better communication and manifested as the cognitive improvement. So the objective of the paper is to evaluate the therapeutic effects of ABTP-MDMP in the brains of Alzheimer's disease in the aspects of first, glucose utilization and metabolism. Secondly, mitochondrial activity and ATP availability. And lastly, synaptic function. The mouse model of Alzheimer's disease, TGCR-ND8, was used for this study, and they received the intravenous ABTP-MDMP twice a week for 2 weeks. So, for example, for PET imaging study, we imaged the mouse on day 0, and from day 3, we started the treatment for 2 weeks. And 3 days after the last treatment, we performed the post-treatment PET imaging to see the effect of our nanoparticles. So brain penetrating polymer lipid nanoparticles were prepared and characterized, and PET-based measurement was used to study the glucose utilization, and electrophysiology test was used to measure the LTP to see the synaptic function, and data was analyzed using the softwares. For the first study using the PET, we were trying to see if the glucose utilization, the glucose use is increased by the nanoparticle treatment. For that, we used the radio-labeled glucose with the PET imaging techniques, and then did the imaging before and after the treatment with two treatment group on vehicle and nanoparticle. As you can see, compared to vehicle control, nanoparticle treatment was able to increase the glucose utilization in the brain across different compartments of the brain. So the effect of nanoparticle was not limited to cortex and hippocampus, which is shown to be important for cognitive function. It was effective in overall regions within the brain. So this shows that either the nanoparticle treatment was able to increase the metabolic activity of the brain so that it uses more glucose, or it would enhance the uptake of the glucose so that the glucose is more abundant on the system. Either way, it's important that the nanoparticle treatment was able to increase the glucose availability and metabolic activity in the brain. So to investigate further on the glucose metabolism and related pathology in the brain, we have used the primary neuronal cell culture to study the mechanisms. So in this case, we have provided oxidative stress in the one group, and we added the nanoparticle alone, which did not show any changes. And lastly, we added the oxidative stress as well as nanoparticle, and we found the protective effect against the oxidative stress in these primary neuronal cell lines. Similarly, we have added the amyloid beta, which is one of the reasons for Alzheimer's disease, and we found that mitochondrial activity is decreased with the incubation, and when we just incubate with nanoparticle, we didn't see much changes. And when we add the nanoparticle with the amyloid beta, we could see the protective effect in its mitochondrial activity. So this was done in vitro, and when we did the in vivo, we were able to see that the mitochondrial amount was increased in the brain homogeneous using the Western blots. And similarly, we found a higher amount of ATP available in the brain with the nanoparticle treatments. So this data suggests that the nanoparticle first increased the mitochondrial activity, and it also increased the energy level, which was shown as ATP availability in the treated brain. And the APTP-MDMP also increased the metabolic efficiency. If you remember, if there is a high amount of oxygen available, the same amount of glucose can be metabolized with higher efficiency to produce the more ATP. Since we saw the more ATP, we thought we would look at the we'll look at the metabolic mechanisms behind it. First, since we saw increased glucose uptake in the brain in the PET imaging, we have measured the glycosylation in the brain, showing the glucose abundance in the system. So we found that increased glucose availability as we expected in the treated animal's brain. And in here, lactate is the substrate that is produced from the glycolysis, anaerobic metabolism, and the nanoparticle treatment was able to reduce the amount of lactate in the brain. Similarly, the lactate transporter expression was decreased with the nanoparticle treatment in both cortex and hippocampus, suggesting that the same amount of glucose would go under the aerobic metabolism to produce higher amount of ATP rather than it goes to anaerobic metabolism, which should produce low amount of ATP. And lastly, it increased the expression of amyloid degrading enzyme, which is neprilysin. Based on the promising effects on the energy level of the brain upon the ABT-PMDNP treatment, we have looked at the synaptic function, which is key mechanism for memory formation and memory maintenance. The results were a little not confirming, but it showed that the nanoparticle treatment may enhance the LTP, which is playing important role in memory formation. Compared to wild type, Alzheimer's disease mouse has shown a distinct difference in this baseline as well as the activation upon the electrophysiological stimuli. Similar to that, we have measured the presynaptic as well as postsynaptic proteins to see if there is any change with the nanoparticle treatment. Synaptophysin, which is presynaptic protein, has increased with the nanoparticle treatment. The postsynaptic protein was increased, however, the significance was not significant enough, although we could see a nice trend with this group. And also PAKT, which plays important role in synaptic function, also is increased with the nanoparticle treatment. And for it to be useful in the clinic, we have to test the toxicity, especially with the long-term treatment. And we found there is no toxicity found in the major organs of the long-term treated animals. And the body weight was consistent, and there is no difference between the treated and vehicle control. In conclusion, ABT-PMDNP improved the glucose availability in the Alzheimer's disease brain, which was shown with the PET imaging. And secondly, ABT-PMDNP protected the mitochondrial function and then aerobic metabolism to increase the cellular energy source, which is ATP. And lastly, ABT-PMDNP improved synaptic function and increased the synaptic proteins, which shows is promising effects on the memory maintenance and cognitive improvements. I would like to thank the scholarship and then grant agencies for this work to be possible. Currently, we are working on different Alzheimer's disease model to validate its effect. And I would like to thank Dr. Wu, Dr. Shirley Wu, who has supervised this work. And if you want to know more about the ongoing projects, please visit our website www.shirleywulab.com to find more information. Thank you. 감사합니다.